Hello everyone, we are Group 4 and for this presentation, we're going to present to you our lab report in experiment number 6 talking about proteins, specifically in color reaction. So proteins are complex macromolecules made up of chains of amino acid that are essential to life and act as the building block of all living things. They participate in variety biological processes including enzymatic catalysis and structural support. Understanding the composition and detection of proteins is essential for various applications in domains like biochemistry, nutrition, and medicine. The goal of this experiment is to investigate protein detection techniques, particularly as they relate to color reaction. Important blood cells constituent like albumin and globulin, which are proteins, demonstrate the critical roles that proteins play in physiological processes like immunological defense and osmotic regulation. Our research is guided by theoretical underpinning that highlights the chemical structure and characteristic of proteins, the capacity of straightforward tests such as Burette, Sanfroprotic, Millions, and Ninhydrin tests to identify proteins based on particular chemical reaction is evaluated. This experiment takes a multidisciplinary approach combining analytical chemistry and biochemistry concepts to fully analyze protein color reaction. Our mission is to advance knowledge of protein analysis techniques and their applicability in various scientific fields by clarifying the underlying theories of these tests and their uses.
For the discussion of methods and materials used or performed in proteins color reaction, the following materials and methods are fresh egg albumin solution, 1% million reagents concentrated nitric acid, ammonium hydroxide solution, Hopkins-Cole reagent, concentrated sulfuric acid, 10% sodium hydroxide solution, 0.5 copper sulfate solution, 0.1 aqueous solution of ninhydrin, dilute alcohol naphtol solution, sodium hydropamide solution, saturated sodium carbonate solution, 2% sodium nitroprusside, and then 1% sodium hydroxide solution. The apparatus used are graduated cylinder, 10 ml of it, and then test tube rack, steering rod, small test tube holder, hot plate, beaker 500 ml, glass droppers, tripod, wire gauze, and water bath. Moving on in the Burette test method is a common biochemical test used to detect the presence of proteins in a sample solution. To perform the test, a small amount of sample is mixed with a solution containing copper sulfate and sodium hydroxide. If proteins are present in a sample, a purple color will develop due to the formation of a complex between the proteins and the copper ions. The ninhydrin test methods involve the reaction of ninhydrin, a chemical reagent, with the amino acid to perform a purple-colored compound known as Ruhemann's purple. The test is performed by applying a solution of ninhydrin to the sample, typically by spraying or dipping a paper strip into the solution. And then Millon's test method, the result of the test provide insight into the individual underlying psychological profile and can be used to help diagnose and treat mental health issues. Millon test method is widely used in clinical settings and has been shown to be a reliable and valid tool of assessing personality traits. Overall, it is a valuable tool for understanding and addressing the complexities of human behavior and psychology. The Hopkins-Cole test method, by observing the color change or other indicators of metabolic activity, microbiologists can use the Hopkins-Cole test to quickly and accurately identify the species of bacteria present in a sample. This method is valuable in settings such as clinical laboratories. And then sulfur test method, used to detect the measure the amount of sulfur present in a sample, whether it is to be a liquid, gas, or solid material. These methods typically involve chemical reactions that produce measurable changes in color or form, allowing technicians to accurately determine the sulfur content of a given sample.
The result.
Today, we conducted an experiment to test the presence of proteins using the Broglie reaction method. We used various samples including albumin, casein, peptone, glycine, and urea to observe the reaction with the Broglie reagent. When we added the Broglie reagent to the albumin, casein, and peptone samples, we observed a color change to violet or purple, indicating the presence of proteins. However, when we added the reagent to the glycine sample, there was no color change, suggesting the absence of proteins. Finally, when we tested the urea, there was no color change upon adding the Broglie reagent, confirming the absence of proteins. In conclusion, our experiment showed that the Broglie reaction method can effectively differentiate between samples containing proteins and those without, providing a simple and reliable way to detect proteins presence.
For the first sample, it is 1% albumin and the reagent used is Millen's reagent. So first it formed a white precipitate after adding the reagent, which eventually turned into a red color after heating the mixture, which indicates the presence of tyrosine, meaning it has a positive result, meaning the sample has protein. The next sample is 1% gelatin, and as observed, even after adding the reagent, shaking the mixture, and heating the solution, it remained with no changes. It remained the same. Therefore, it indicates the absence of tyrosine and a negative result. For the sample 1% casein, after adding the Millen's reagent, the solution formed a white precipitate. Then, after shaking it and heating the mixture, it turned into a red color, which implies that there is a presence of tyrosine and it has a positive result. For the second table and the sample 1% phenol, it formed a clear precipitate after adding the reagent, but after shaking it and heating the solution, it showed change in color and it turned into a dark orange precipitate, indicating the presence of tyrosine, meaning it has a positive result. Starting with the result with number 1, so for the first table, the sample albumin and casein had the same reaction, forming a red precipitate, indicating the presence of tyrosine and a positive result. However, the sample gelatin showed no change and no reaction even after adding the reagent, implying that there's an absence of tyrosine and it has a negative result. But for the sample phenol, it shared the same reaction with the samples albumin and casein, having the formation of a different color precipitate after heating the solution. However, for the sample phenol, it turned into a dark orange color, while albumin and casein had a red precipitate. But still, it has a positive result due to the presence of tyrosine.
In this section, the Santoprotein test is conducted to detect the presence of aromatic amino acids in various protein samples. So the test starts with a series of samples, and we'll be observing the color changes after adding nitric acid and ammonium hydroxide reagents. In sample number 1, 1% albumin, upon adding the reagents, we observe a yellow to orange color formation. This indicates the presence of aromatic amino acids like phenylalanine or tyrosine in albumin. In sample number 2, 1% gelatin, we notice either no color change or slight yellowing. This is because gelatin consists mostly of non-aromatic amino acids, resulting in a weak or absent reaction in the Santoprotein test. In sample number 3, 1% casein, similar to gelatin, casein also shows no significant color change. It's predominantly composed of non-aromatic amino acids with only trace amounts of aromatic ones, leading to minimal reaction in the test. In sample number 4, 1% phenol, we see a yellow to orange color formation akin to albumin. This confirms the presence of aromatic compounds, indicating the presence of aromatic amino acids. Comparing the results of samples 1, 2, and 3, we observe that the presence of aromatic amino acids determines the intensity of the color change in the Santoff proteomic test, while albumin and phenol exhibit a pronounced color formation due to their aromatic structures. Gelatin and casein show limited or no reaction due to their non-aromatic composition.
So, test for sulfur. The sample is 1% albumin. The reagent added is NaOH plus Pb C2H3022. So, the result is the presence of sulfur containing amino acids is indicated by the formation of a black precipitate. The observation or discussion, the development of a black precipitate will be the finding. Sulfur can be detected in the sample by looking for this dark precipitate. Thus, the presence of sulfur in the sample is indicated if a black precipitate forms in the solution containing 1% albumin after adding NaOH and PbC2H3022. The second sample is gelatin, and the reagent added is NaOH plus Pb C2H3022. The result is no black precipitate is observed, suggesting the absence of sulfur in amino acids in gelatin. And the observation or discussion is formation of a black precipitate. The third or last sample is peptone, and the reagent added is same, NaOH plus Pb C2H3022. So the result is when a black precipitate forms, there is cysteine present in the solution. And the negative result is the absence of black residue suggests a deficiency in cysteine. And the observation or discussion is same in gelatin, formation of a black precipitate. So, compare the result with number 1. A positive result is seen for albumin and peptone, which combine with sulfur amino acids to generate a black precipitate. It is possible that gelatin lacks sulfur amino acids since it does not produce a black precipitate.
For Hopkins Cole reaction on our first sample, the 1% albumin, when we added the concentrated sulfuric acid, it may result on forming violet ring. Why? It is where the tryptophan test is positive. A purple ring will appear between the two layers. This occurs as a result of tryptophan reaction with glyceric acid and sulfuric acid that has been concentrated, and it produces the purple ring. While on 1% gelatin and 1% cation, when we added the concentrated sulfuric acid, it may no change at all.
Performing an anhydrine reaction method. When 0.2 albumin and 0.1 anhydrine solution are combined, the albumin proteins react with anhydrine to form a purple complex. Overall, the combination of 0.2 albumin and 0.1 anhydrine solution offers a simple yet effective method. When ammonia 0.1 anhydrine solution is added to a sample, it reacts with an amino acid present in the solution. The anhydrine reagent specifically binds to the amino acid, performing a red-colored compound. When urea 0.1 anhydrine solution is added, it reacts with the urea to form a purple-colored complex. And then within anhydrine is a compound that reacts.
Question number 1. Why do all proteins give a positive Breuer test? Today, we explored why all proteins yield a positive Breuer test. It turns out that the peptide bonds, a fundamental structural element of proteins, are the key. When proteins are subjected to the Breuer test, the peptide bonds react with the copper ions present in the Breuer reagent, forming a complex. This complexation reaction results in the distinct color change, typically color violet or purple, indicating the presence of proteins. Because all
All proteins are composed of peptide bonds. They are consistently produces a positive Broyat test result, making it reliable method for protein detection.
Why does nitric acid stain the skin with yellow color? Because nitric acid oxidizes, it can leave a yellow stain on the skin. Nitric acid can react with the proteins on the skin surface when it comes into touch with the skin, changing the chemical composition of the skin and leaving it discolored a yellow color. When nitric acid is concentrated or comes into prolonged contact with the skin, the staining effect becomes more apparent.
What grouping in amino acids or proteins is responsible for ninhydrin reactions? Amino groups NH2 found in amino acids and proteins are the main target of the ninhydrin reaction, a colored chemical known as Rohemann's purple, which is usually purple-blue in color, is formed when ninhydrin combines with certain amino groups. This reaction offers visual signal of the presence of amino acids or proteins, which makes it useful in biological experiments for their detection and quantification. Will methionine give a positive sulfur test? So the answer is yes. A positive sulfur test can be obtained with methionine. A positive sulfur test can be obtained by reacting methionine, an amino acid that contains sulfur, with certain reagents. When looking for sulfur containing chemicals in organic molecules, this assay is frequently utilized.
For question number 5, what color will be produced by proline with ninhydrin and why? So the color produced by proline and its reaction with ninhydrin is the color yellow. Amino acids mostly react with ninhydrin, but due to the complex of its chemicals compositions and chemical structures of proline and its amino acids, it reacts differently than most of them. Ninhydrin reacts and forms a purple color with primary amines. However, proline is a secondary amine, hence the formation of a yellow color. To conclude the experiment of proteins in reaction with color, the tests such as Biret test, Ninhydrin test, Santroproteic test, Millen's test, and Hopkins-Cole test are experiments or tests used for detecting or determining the presence or absence of proteins in the solution or sample. The certain reagent used for the test will determine whether or not the sample contains proteins. The reaction of the experiment will result to a change in color if it is positive, but if it is negative, there will be no changes.
So here is our reference for experimental number 6. So that's it, everyone. Thank you and have a great day to you all, and may God bless you.